Welcome to Brilliant Biology. Today we're diving into topic 5b6, investigating abundance and distribution of organisms. By the end of this video, you should be able to define abundance and distribution, you should understand the three types of distribution, and you should also be able to describe the ecological techniques that are used to measure abundance and distribution. Let's begin by looking at the definitions of abundance and distribution. Abundance is the relative representation of a species within a particular ecosystem. So let's take a sheep for example. Abundance is not just the number of sheep in a field, but also the number of sheep in the field compared to the number of other organisms in that field. Distribution, on the other hand, is where a species of organisms is found in the environment and how it's organized. Some organisms are found in very high densities, such as ants in a nest, and others are found more spread throughout the habitat, such as top predators. Distribution can also change because some organisms, such as birds, will migrate to different countries or different areas at different times of the year. The overall difference between abundance and distribution is that abundance is looking at the numbers of organisms, whereas distribution is looking at how these organisms are arranged in a geographical space. Now let's look at the three main types of distribution. The first one is uniform distribution. As you can see by the image, this means that the organisms are evenly spread throughout. This happens when resources are thinly but still evenly spread, or when individuals of the same species are hostile towards each other. Moving on, we have clump distribution. In this type of distribution, we can see organisms organized in groups. This happens when animals or plants have specific resource requirements, and so they group together in areas where those resources are found. And finally, we have random distribution, and as you can see, that means the organisms are randomly scattered with no real pattern, and this happens when there's plenty of resources and no hostility. Now let's look at ecological techniques that are used to measure abundance and distribution. The first one is quadrats. A frame quadrat is a square frame that is divided into sections, and you lay it on the ground to identify the sample area. You can measure the individual counts, which is the number of individual organisms in an area, but you could also measure the percentage cover, which is the area that is covered by a particular species. And these results can then be used to calculate a mean for the abundance and distribution. Now let's explore the advantages and limitations of frame quadrats. They have a lot of advantages. The first one is that they're useful for plants and animals which don't move much. They also come in a variety of sizes and they're easy to handle. On the other hand, there is a limit to the size of the area that you can sample, and you have to make decisions about including or excluding organisms that are only partly touching the quadrat. Lastly, there is an issue with randomness. You do want the sampling to be random because you don't want to be biased and only focus on the most interesting parts of an ecosystem, and now you have information that doesn't accurately reflect the entire ecosystem. But at the same time, simple random sampling could be an issue because you could miss out on important organisms, and then we also don't have an accurate picture of the entire ecosystem. Next up, we have a point quadrat, and this is a horizontal bar that is supported by 2 or 3 legs. At set intervals, there are holes through which these pins are dropped, and the species which touch these pins are recorded. Then we have permanent quadrats, and as the name suggests, they're left in a place permanently, and they measure the changes over the seasons or over the years. The big advantage with them is that they have reliable data. Lastly, we have aerial quadrats, and these are aerial photographs of very large areas of land, and they're then divided into areas so they can be surveyed for species abundance and distribution. They're often used for very large areas that are hard to reach, such as deserts or savanna. One way of measuring the abundance of organisms with a quadrat is using the ACFOR scale. In this scale, A stands for abundant, C stands for common, F for frequent, O for occasional, and R for rare. But obviously there's limitations to the scale. First of all, it's very subjective because 2 people would never give the same exact scores for the same species. There's also no set definition for the terms. so there's no number of organisms that would be classed as abundant or common or frequent or so on. And lastly, species can easily be rated according to how obvious they are rather than how frequent they are. Finally, let's look at transects. A line transect is a tape that is stretched out between 2 points, and we record the organisms that touch that tape. A bell transect is 2 tapes that are stretched out, and the area between those 2 tapes is surveyed. We could also have 1 tape be stretched out and quadrats taken at regular intervals to have the same effect. You have to remember that transects are not random, they're systematic, and we can use them to investigate a change in distribution of organisms linked to a change in an abiotic factor. So for example, we could see if there's a change in the distribution of organisms as we get closer to or further away from a water source. To finish off, let's look at measuring abiotic factors. You have this table in your book and it summarizes the different abiotic factors and what methods are used to measure them. Scientists also use data loggers, which are basically devices that can measure several abiotic factors. And that is the end of this video. Thank you so much for watching. If you have any questions, just leave them in the comments. And if you enjoyed this and if this helped you at all, please leave a like and subscribe. Thank you so much.